Hey folks, it's Mike here with another amazing new product. That's right folks, it's the Cell Chop. You're going to be in a great mood all day because you're going to be slapping your cell division problems away. And I'm here with Jenny Ghosh, say hello.
Hi.
An expert on cell division. Alright folks, first off, make sure your Cell Chop is in mitosis mode. Give it one slap and it's an interface. Tell them what happens, Jen.
Sure, Mike.
This cell is an interphase, which consists of G1, S, where your DNA replication takes place, and G2. Most of the time, the cell in interphase is resting, or carrying out normal function.
Alright folks, enough of the boring stuff. Slap your Cell Chop twice more, 1, 2, and your cell is now in prophase, prometaphase. And Jen here is going to tell you what happens.
In this phase, the chromosomes condense and become visible, and the nuclear envelope disappears. Don't forget that the centrosomes move apart and the mitotic spindle begins to form.
But come on, folks, who really cares about a Houdini wannabe nuclear envelope? Slap your Cell Chop 3 more times, 1, 2, 3, and your cell is now in metaphase. Your chromosomes are going to be lining up like they're best friends. Take a look at this.
In metaphase, the chromosomes are aligned on a metaphase plate and the mitotic spindle forms. Kinetochore microtubules are connected to the kinetochores on each sister chromatid so that the chromosomes are ready to split.
Alright folks, these cells do not like each other though because they're splitting up with 3 more chops from the Cell Chop. Count them, Jen.
1, 2, 3.
Bam, now your cells are in anaphase.
In anaphase, the length of the mitotic spindle shortens and the chromosomes split, now making double the amount of chromosomes. This ensures that each daughter cell gets identical sets of chromosomes. Now in telophase, chromosomes decondense and the nuclear envelope reforms. In cytokinesis, when the cytoplasm divides, 2 daughter cells are now formed with the same amount of chromosomes and genetic information as the parent cell.
Cell Chop over once, and it switches to meiosis mode. And with one slap from your Cell Chop, tell them what happens, Jen.
Well, I'll tell you, Mike.
In prophase 1, the chromosomes condense and become visible, centrioles form and move toward the poles, and the nuclear membrane begins to dissolve. Homologs pair up, forming a tetrad, which is comprised of 4 chromatids. The homologous chromosomes will swap genetic material in a process known as crossing over. Crossing over serves to increase genetic diversity by creating 4 unique chromatids.
Transition from prophase 1 to metaphase 1, almost as exciting as this twig that looks like pie.
With 2 more slaps on the Cell Chop, you're in metaphase 1, where microtubules grow from the centrioles and attach to the centromeres. In this phase, the tetrads line up along the metaphase plate.
So with 2 more slaps on your Cell Chop, your cell goes into anaphase 1. But what happens in anaphase 1? We're going to tell you.
Now in anaphase 1, the centromeres break and homologous chromosomes separate. Cytokinesis begins. Don't forget, cytokinesis is when the cytoplasm splits.
You're almost done with meiosis 1. With 2 more slaps on your Cell Chop, your cell goes into telophase. But what happens in telophase, Jenny?
Let's see.
Telophase 1 is when the chromosomes start to decondense. In this phase, cytokinesis reaches completion, creating 2 haploid daughter cells.
Spin the Cell Chop around 180 degrees and it switches to meiosis 2 mode. Let's get the expert's opinion.
Meiosis 2 is just as important in the cell division process as a dim light is in a dark room.
I still can't see anything.
In this phase, the centrioles form and move toward the poles and the nuclear membrane dissolves.
In metaphase 2, which is very similar to metaphase 1, the microtubules grow from the centrioles and attach to the centromeres. The sister chromatids line up along the metaphase plate.
Alright folks, so we just finished up some metaphase 2. With 2 more slaps on your Cell Chop, your cells are going to go into anaphase 2. Go ahead, Jen. But what's the difference between anaphase 1 and anaphase 2? We're going to let you know.
Anaphase 2 is when the centromeres break, sister chromatids separate, and cytokinesis begins. In telophase 2, the chromosomes may decondense and cytokinesis reaches completion. This creates 4 haploid daughter cells, and each of these cells contains half the number of chromosomes than the parent cells. Mitosis and meiosis are important because without cell division there would be no reproduction of living things. Almost every living thing that is alive originated from single-celled organisms, and without mitosis, these organisms would not have been able to reproduce. Without meiosis, genetic variation wouldn't exist at all.
Hot babe walks by, you not know what to do? Use the Cell Chop. Get that massive pump you need right before the girl passes by. Be like, what's up? Cell Chop, use it.
I know you want to look like this, so if you want to look like me over here, get the Cell Chop. Remember that name. And maybe one day you'll look like the Broski.